Methylation-specific MLPA, or MS-MLPA, is an adaptation of the targeted MLPA technology and combines CNV detection with DNA methylation profiling. In this video, typical MS-MLPA results are shown, and I will explain how they can be interpreted. Note that, for simplicity, the following applies to the cases used in this video. The methylation-specific probes target imprinted genomic sequences, and the reference samples have 2 copies for each probe target. More complex situations and explanations exist, depending on the used probe mix. However, as we want to demonstrate the general principle, these cases fall outside the scope of this video. The analysis of MS-MLPA data is divided into 2 parts. In the first part, copy numbers are determined by normalizing the undigested test samples against the undigested reference samples, which results in copy number ratios, similarly to a standard MLPA analysis. In the second part, the methylation status of a sample is determined. This is done by comparing the digested reaction of a given sample to its undigested counterpart. This will give you methylation ratios. The reference samples determine which results are considered normal. The calculated methylation ratio of a methylation-specific probe indicates how many copies are methylated in a sample. Therefore, you first need to know the copy number of a probe to understand its methylation status. To see how this works in practice, I will give an example and explain how to interpret the results. If you are unfamiliar with how MS-MLPA data is analyzed and results are displayed by Coffeelyzer.net, please refer to Appendix 1, Normalization and Result Interpretation of the Coffeelyzer.net reference manual. During the analysis of MS-MLPA data, we first determine the copy number and then the methylation status. The results of the copy number analysis are displayed in the upper chart and of the methylation status analysis in the lower. In this example, we will have a look at the results of a sample obtained from a normal, healthy individual. As expected, the probe ratios in the undigested sample are similar to those in the reference samples. Because the reference samples have 2 copies for the target sequences, this means that this sample has 2 copies as well. Now that we know the copy number, we turn to the digested sample. A methylation-specific probe can only generate a signal when its target sequence is methylated, so a methylation ratio of 0.5 means that 50% of the target sequence of a probe is methylated. As this particular sample has 2 copies and a methylation-specific probe's a methylation ratio of 0.5, the number of methylated copies in this sample is 1. Given the fact that this sample is derived from a healthy individual, it is no surprise that the results are similar to those of the reference samples. Several cases will now be presented, showing a variety of possible results, followed by an explanation. If you want, you can test yourself and try to find an explanation for the results of the sample in question before the timer has finished. Of course, you can always pause the video if you need more time. Now we look at sample 2. This sample also has 2 copies, as can be concluded from the undigested sample results. But now the ratio of the methylation-specific probes is 0, indicating that both copies were effectively digested by HHA1 and were therefore not methylated. Similarly to the previous two samples, sample 3 also contains 2 gene copies. When looking at the digested sample, however, we can see that the methylation-specific probes have ratios of approximately 1. This means that both gene copies were protected from HHA1 digestion and were thus methylated. The undigested sample 4 shows copy number ratios of around 0.5, indicating that only one gene copy is present. The methylation ratios being 0 in the digestion reaction indicate that the probes could not be amplified. This means that the single copy that is present in this sample is unmethylated. Based on the ratios of 0.5 in the undigested sample, we can conclude that sample 5 contains one gene copy. The methylation ratios in the digested sample are close to 1, meaning this single copy was not digested by HHA1 and is thus methylated. Sample 6 shows probe ratios of a value close to 2 in the undigested reaction, indicating a copy number of 4, twice the normal value. In the digested counterpart reaction, the methylation-specific probes have ratios around 0.25. This means that 25% of the gene copies present is methylated, which, in this sample, is one copy. Sample 7 is an interesting one, as the methylation status is different for two subsets of probes. Before we get to that, we first need to determine the copy number. All target probes have a ratio around 1.5 in the undigested sample, meaning 3 copies are present. As you can see in the digested sample, there are 2 groups of probes with different methylation ratios. This means the number of methylated copies is different for both these groups. The first group has a ratio of around 0.3. This comes down to 1 methylated copy and 2 unmethylated copies. The methylation ratios of the second group lie between 0.6 and 0.65. So for these targets, the situation is the other way around. 2 copies are methylated and 1 copy unmethylated. For a genomic condition, such a result would be very unusual. But results like this are not abnormal when looking at the methylation profile of tumors, for example. The methylation profile of a sample cannot be interpreted without knowing the copy number. It is therefore essential first to interpret the results of the undigested sample to establish the copy number. Hereafter, you can look at the results of the digested sample to determine its methylation status.